Okay. Good evening, everyone. Last week for the 2026 class, we had an extra class, so we discussed mitosis over there. And now I have decided to have a free webinar on meiosis since it's equally important for the 2025 students as well, right? And today we'll be discussing, meiosis, right? Today we'll be discussing meiosis. So during this discussion, you need to have your resource book with you, right? Because whenever I ask you to write something down, for example, sometimes I can give you tips, right? Sometimes I can ask you to write down the questions, right? For questions, I'll tell you what to do separately. For now, you need to have your resource book with you because if I give you some annotations to be written, during the time that we are doing the lesson, you need to quickly write down in your resource book. Plus, during the class, that means when I'm teaching you, if you think that this particular thing that I told you is important for you, then you should again write that down in your resource book, right? So everything depends on you. You need to write down everything that you need to write down the important things I said, plus the important things that you believe will be important for you, right? And also for the 2026 students, they have their writing book with them. So if I give you questions, you all can have a piece of paper. So if I give you questions, you can write them down. And these questions are not to be studied, right? These questions are given. If you want, you can study, but these questions are given so that you all are engaging with me during the lesson, right? Now, usually I do not give much questions in the official classes, but since this is a free webinar, I'm giving you questions to keep you engaged during the lesson, right? Plus, usually we give the weekly bundles and the theory class. So those things will be given after the class is over. Probably tomorrow it will be given to the official class students plus the 2025 official class students, right? Okay, so without further ado, let's start the lesson. Now I will be sharing my screen. If you have any doubts, you can always WhatsApp me through your personal number. I'll be always checking the WhatsApp, right? You can WhatsApp me any of your doubts, right? Okay. Right. So hope everyone can see the screen. Okay. So last week we discussed mitosis. When we discussed mitosis, I hope that you all can remember the interphase, right? The interphase is equally important for meiosis as well, right? Okay, so let's read what this meiosis is. But before that, I should tell you something. That is, in mitosis, what did we learn? In mitosis, can you remember, we learned it gives rise to two genetically identical cells, right? The end products are two genetically identical cells, right? That means the two daughter cells produced are identical to each other as well as they're identical to their mother, right? So that is what happens during mitosis. In meiosis, actually the opposite thing happens, right? In mitosis, genetically identical offsprings were given, right? But in meiosis, genetically non-identical offsprings are given, right? In meiosis, genetically non-identical offsprings are given. And also there is one major difference. In mitosis, we saw two daughter cells are produced, right? But at the end of meiosis, you will see that there are four daughter cells produced, right? Four daughter cells produced, okay? And also, can you all remember when we did mitosis, mitosis had the largest phase called the interphase, right? It had the largest phase called the interphase, right? Just likewise, in meiosis also, it has the interphase, right? It has the interphase. And after interphase in mitosis, can you all remember we came across prophase, metaphase, anaphase, and telophase, right? Anaphase, telophase, and cytokinesis. Just likewise, in meiosis also, we have these 4 phases, but we distinguish them using some different numericals, right? We use some numericals to distinguish them, right? Okay, so that is the brief idea about mitosis and about meiosis. Now let's come to the reading part, right? In meiosis, they are saying sexually reproducing organisms undergo different types of cell division called meiosis. Can you all remember when we did mitosis? Under that, we learned an importance of mitosis is asexual reproduction. Why? Because in asexual reproduction, you need only one parent, right? Not a human, actually. Let's take a grass, right? You need only one parent, right? From that parent, identical offsprings can be produced, right? That is asexual reproduction. For that, mitosis was important. For meiosis, that means meiosis is important in sexual reproduction, right? In sexually reproducing organisms, can you all see genetically identical offsprings? Apart from twins, can you all see genetically identical offsprings? No, right? They're not even identical to their parents, right? They have a mixture of the 2 parents, right? Now, a copy of the daughter is not exactly similar to the mother, right? A daughter has the genes from both mother and father, right? That is due to this meiosis. Actually, our gametes, what are gametes? Gametes are actually in sexual reproduction, the females produces eggs and the males produces the sperms, right? These are called gametes, right? These are the gametes. Gametes are the vehicles that carry the chromosome, in order to form a zygote. A zygote. What is a zygote? A zygote, you might have heard this before in your O-levels, when a sperm and when an egg meets with each other, it forms a zygote, right? It forms a zygote, right? So basically, sperm is produced by meiosis, egg is also produced by meiosis, right? And now we are going to learn how it is being produced, right? Okay. Sexually reproducing organisms undergo different type of cell division called meiosis. All right. Meiosis is a type of nuclear division which give rise to 4 haploid, genetically non-identical daughter nuclei from a diploid mother nucleus. Right. In mitosis, what happened? A diploid mother cell, that means a 2N mother cell, gave rise to a 2N daughter cell, right? 2N. What do you mean by 2N? 2N, that means there are 2 pairs of chromosomes. For example, in humans, how many chromosomes are there? 46 chromosomes are there. In humans, 46 chromosomes are there, right? We know they are in pairs. That means 23 pairs of chromosomes are there. Since they are in pairs, we call them as diploid, right? Diploid. Since they are in pairs, we call them as diploid. The 23 chromosomes, when I did mitosis, I drew the 23 chromosomes, the 23 pairs of chromosomes, and showed you, right? That they are in pairs like this, right? So therefore, they are diploid. What happens in mitosis is the same diploid condition is produced again, right? The same diploid condition was produced again. There was no change in the diploid. That means there was no change in the ploidy level, right? Ploidy level. Ploidy level means whether it is diploid, whether it is triploid, right? Triploid means there are 3 sets of chromosomes like this, right? Triploid means you will come across these ploidy levels under molecular biology when you go to grade 13, right? You don't have to worry about the ploidy levels, right? So basically, diploid means having pairs of chromosomes like this, right? Like humans, right? We are diploids, right? So our gametes, if you look at our sperm, if you look at our egg, They are not diploid, they are haploid, right? If you look at us, all our cells, right? All our cells, even our ovaries, even our testicles, right? Even they, all of them are actually diploid. But the gametes that they produced, you know, ovaries only produce the eggs, right? In humans, I'm talking, okay? Ovaries only produce the eggs, right? Ovary is 2N, it is diploid, but the eggs that it produces is actually N, right? And if you take the testicles, again it is 2N, but the sperms it produces, sperms are produced inside the testicles. The sperms which are produced is N. N means haploid, right? N means haploid, 2N means diploid, right? So what is this? Why are they haploid? Okay, we will learn why they are haploid. After we finish discussing meiosis, you will understand why they become haploid, right? But you have to understand at the moment, in our body there can be diploid cells. All our cells are diploid except for our gametes, right? Our gametes are haploid cells. Okay, our gametes are haploid cells because they have been arisen from, that means they have formed from meiosis, right? Their mother cell was there and the mother cell started dividing using meiosis. Mother cell was 2N, but at the end, the 4 daughter cells produced from one mother cell all were haploid, because you know in meiosis I told you 4 daughter cells are produced and all of them are haploid, right? This was 2N, but this was N, right? That means haploid means what? They have only one set of chromosome, right? Just one set, right? Just one set. That means now in humans, how many pairs of chromosomes? In humans, our normal cells have 23 pairs of chromosomes. That means how many chromosomes? 46 chromosomes. But if you take the egg produced inside the human, the egg produced inside females, how many chromosomes does it have? It just has 26 chromosomes, right? 26 chromosomes, no pairs, right? 26 chromosomes. Since it doesn't have pairs only, we call it haploid, right? So I wanted to tell you the difference between diploid and haploid. Hope you understood that part, right? Okay, so they're saying meiosis is a type of nuclear division. Why are they referring to nuclear division over here? It's because it's because in mitosis also we learned mitosis under mitotic phase. Can you remember? Mitotic phase has 2 parts. What are the 2 parts? Mitosis, and the other part is cytokinesis, right? Mitosis, and the other part is cytokinesis. Mitosis is the nuclear division. Cytokinesis is the cell division. In mitosis, the nucleus divides, and in cytokinesis, the cell divides. Just likewise, in meiosis, the nucleus divides. Then after following meiosis, you again have cytokinesis. In cytokinesis, the cell will divide, right? So that's why we refer to meiosis as a type of nuclear division which give rise to 4 haploid genetically non-identical daughter nuclei, right? Type of nuclear division which give rise to 4 haploid genetically non-identical daughter nuclei from what? From a diploid mother nucleus. The mother nucleus is diploid, but the daughter nuclei which are formed are haploid, right? This is a SCQ point, right? A SCQ point, right? Meiosis involves 2 consecutive nuclear divisions. Meiosis 1, meiosis 2. Now, when we learned mitosis, in mitosis, we didn't learn we didn't divide mitosis into mitosis part 1, mitosis part 2, right? We just learned mitosis as a whole process, right? But in meiosis, it has been divided into 2 consecutive nuclear divisions. That means how many nuclear divisions are taking place inside the meiosis process? Not inside, in the meiosis process, how many nuclear divisions are taking place? 2 nuclear divisions are taking place, right? The first nuclear division is called meiosis 1. Second nuclear division is called meiosis 2. Right. Meiosis 1 is the reduction division. Why do you call meiosis 1 the reduction division? That is because in meiosis 1 only, the 2n gets converted to n. In meiosis 1 only, 2n gets converted to n. You can see there is a reduction, right? There is a reduction. That is why you call it the reduction division, right? The diploid cells become haploid cells, right? At the end of meiosis 1, you will have 2 cells, right? At the end of meiosis 2 only, you will have 4 cells, right? At the end of meiosis 1, you will have 2 cells. At the end of meiosis 2, you will have 4 cells, right? If you have any doubts, write them down and then later ask me after I fully discuss the meiosis part, right? If you have any doubts on this part. Now, because this part cannot be understood, sometimes it will be difficult for 2026 students to understand without fully studying the whole process, right? Okay. Meiosis 1 is a reduction division and meiosis 2 is similar to mitosis. Okay, now this is an important point. If they ask you in which meiosis division is similar to mitosis, then you should be able to write it's meiosis 2, right? We will learn the similarities between mitosis and meiosis 2. Mitosis and meiosis 2. Remember, similar does not mean same. Okay, same and similar in English also it has 2 different meanings. Similar means in the process is sort of similar, but there are some differences as well. We will see what those differences are as well, right? Each stage consists of 4 subphases. That means in meiosis 1 as well as in meiosis 2, you will find some subphases, right? They're saying they are prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. But in mitosis, can you remember, there was another phase called prometaphase. Prometaphase, I told you, it's between prophase and metaphase. That's why you call it the prometaphase, right? But meiosis is a bit easy when compared to that, because in mitosis, you will have to remember everything. You have to remember these things in order. Last week also I told you, right? You have to remember all these processes in a particular order. First, this thing happens, then this thing happens. Likewise, you have to remember it in a particular way, right? Order, right? In prometaphase, you have to remember specifically what happens in prophase, then specifically what happens in prometaphase. It'll be a bit difficult to study the different, that means to study them separately. But what if prophase and prometaphase join together and form just prophase in meiosis, right? Prophase and prometaphase join together and just form prophase. Prometaphase is also included in the prophase phase. So it's easy for you to learn everything, right? Because you know, the nucleolus disappears, the nuclear envelope fragments, right? We learn these 2 things under 2 separate phases in mitosis, if you can remember, right? But in meiosis, we learn both these things under prophase. Therefore, it will be easy for us to study. Okay, so there is one advantage. Even though this is big, you do not have to worry about this, because in 2022, they asked about meiosis 1, right? Meiosis 1. So the remaining ones are mitosis and meiosis 2, right? So meiosis 2 and mitosis, they are the remaining ones that could come for unit 2 essays, either for 24, 25, or 26, right? 2026 students, right? Before meiosis, one cell is in interphase. During S phase of the interphase, DNA replication occurs. Right, so all these, that means either meiosis, mitosis, everything starts from one cell, right? Everything starts from one cell. This particular cell actually has to undergo interphase, like I told you. In interphase, how many subphases are there? G1 phase, S phase, and G2 phase, right? In G1 phase, proteins essential for S phase were synthesized. In G2 phase, again, proteins were synthesized, and you can remember centromeres, centrosomes duplicated, right? Centrosomes duplicated. That was a unique feature feature that happened, that was a unique feature present in G2, right? And in S phase, what happened? I told you in S phase, DNA replication occurs, right? For the 2026 students, if they were present for the if they were present last week, then you all can remember I told you the clear difference between duplication and replication, right? So basically, now they both have the same meanings, but the word duplicate is a bit different from the word replicate, right? So if you take DNA replication, for 2025 students also, I will explain it, right? Now, DNA replication and DNA duplication. Now, if I take this word, right? If I take this word duplicate, right? If I take this word duplicate, and see, I duplicate. Can you see an exact same word was produced, right? In DNA replication, you will learn this in molecular biology as well. Something different happens, right? In DNA replication, you know, DNA is a double-stranded molecule like this, right? DNA is a double-stranded molecule like this, right? What happens in DNA replication is these strands get separated. Then a new strand is formed, right? A new strand is formed. At the end, you can see, at the end, actually, these two are also similar to this. Okay, these two which are formed are also similar to this. But actually, what happens is, you can say it's not a duplication which takes place. It's a replication, right? At the end, all three DNA molecules are same actually. But you can see duplication word and replication word has a difference, right? It's just a difference. That means it doesn't make a big impact, right? They won't ask you the difference between duplication and replication, right? Okay, so now, now I was talking about S phase. In S phase, we know DNA replication takes place. That means now it's like this. Our chromosomes are like this, you know, it's entangled in a mesh, sort of like thingy in S phase, right? In S phase, you know, it appears as sort of like a mesh sort of thingy. But I told you clearly, in prophase, in mitosis, still I didn't come to meiosis. Prophase, in mitosis, they become condensed, right? The chromosomes become condensed and they appear with two sister chromatids, right? They appear like this, right? The chromosomes appear like this in the prophase in mitosis. If you all can remember, right? In mitosis, the chromosomes appear like this. That is because they were replicated in S phase. They were replicated. Replicated means, can you see? This part is exactly similar to this part. They were replicated, right? The DNA inside the chromosomes were replicated. If they haven't replicated, they would just look like this, right? In prophase, after condensation takes place. What is condensation? Condensation does not mean the condensation in chemistry, changing from water vapor to liquid. Here, condensation means becoming condensed. Condensed means sort of like concentrated, sort of thickened, right? Here it means becoming thickened, right? So you can see over here that in prophase, you can see the chromosomes are getting thickened, right? Or condensed, right? What if they were not replicated? They would appear like this, right? Before prophase, they appeared like a mesh like this, right? They appeared like a mesh like this, right? Tangled together, right? You can't even clearly see them in the light microscope. In prophase only, you can see the chromosomes clearly in the light microscope, right? And what happens? What if they, now I'm telling you, in prophase, because they were duplicated in S phase only, because they were replicated in S phase only, you can see structure like this, right? You can see two structures like this, right? Two structures. If they were not replicated in prophase, they would have appeared like this, right? The chromosomes would have appeared like this. Okay, so like that, right? So in there is that now you know that in if in S phase only the replication takes place. The replication plays a major role. If DNA replication did not take place, we can't carry out mitosis, we can't carry out meiosis, right? Because in mitosis, if DNA, now when DNA replication takes place, if 46 chromosomes were actually, if 46 chromosomes were present, actually 92 chromosomes will be there. 92 chromosomes will be there due to the DNA replication, right? 92 chromosomes will be there due to the DNA replication, but they appear like an attached structure like this, right? Attached structure like this, right? So you can see why it is important. After that, only the 92 chromosomes are again divided into the two cells, like 46, 46 to form two identical daughter cells, right? So you can see why DNA replication is important, right? Okay, so you can see before meiosis, one cell is in interphase. One cell means meiosis starts from a cell, right? So during the S phase of that interphase, DNA replication occurs, right? And in G1 phase, proteins are synthesized, right? In G2 phase, again, proteins are synthesized, right? The same things happens, right? Okay, now we are moving on to meiosis 1. Now you can forget about mitosis. Now we are coming into meiosis 1. In meiosis 2, let's think about mitosis. But for meiosis 1, it's a completely new thing, right? Let's see. Cell enters to the prophase from the interphase, right? So we know that after interphase only the prophase comes. So cell enters to the prophase from the interphase. Chromosomes begin to condense. What do you mean by condense? Becoming thickened, right? Becoming thickened, right? They become shorter and they become thicker, right? Becoming condensed. That means becoming concentrated, right? Looking concentrated, right? Chromosomes begin to condense. That is the first point, right? This order is very important. First thing which happens is chromosomes become thickened. That is the first thing. Then what is the second thing? Second thing is nucleolus begins to disappear. What is the nucleolus? Nucleolus is the small structure inside the nucleus, right? That disappears. That is the second point, right? First, chromosomes begin to condense. Second, nucleolus disappear. Next, formation of zipper-like structure called the synaptonymal complex by a specific proteins holds the two homologous chromosomes tightly together, right? Holds the two homologous chromosomes tightly together. What are homologous chromosomes? Last week also I explained it to you, but this week also I'll explain it again, right? For this to be a complete discussion, I will explain, right? Okay. Okay, okay, 1, 2. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 1, 2, 3, 4, 5, 6, 7, 8. Okay. Right. Okay. Now, okay, it's like this. Now, 1, 2, 3, 4, 5, 6, 7, 8. All right. So there are 8 pairs. Now let's take this is a certain animal. That certain animal has 8 pairs of chromosomes, right? 8 pairs of chromosomes means what? How many chromosomes? 16 chromosomes are present, right? In this particular animal, 16 chromosomes are present, right? This is in the unreplicated form. That means before replication, this is how the chromosomes look, right? After replication, this is how the chromosomes look.
right? Before replication, it looks like the first box. After replication, it looks like the second box, right? You can see, so basically we can in prophase we see after replication, right? In prophase we see after replication, so we see the second box chromosomes, right? Before replication we can't see the structure of the chromosome. We can't see the structure of the chromosome because they are in a mesh sort of thingy, right? That means you can't clearly see them, they are entangled, right? All these chromosomes are entangled, so you really can't see the structure. But this is the real structure of the chromosomes before replication. After replication, you can see still 8 pairs of chromosomes are there, but can you see there are 2 strands like this, right? That shows the replication, right? That shows the replication. Okay, so what are the homologous chromosomes, right? Now, can you see I have drawn them in pairs? These I have drawn all these chromosomes in pairs, right? One is from the mother, one is from the father. Again, one is from the mother, one is from the father. Just likewise, in this set also, this is the same set, just the replicated set. Mother, father. The mother and the father chromosomes are identical to each other. I have tried my best to draw the same structure over here. See, they both are identical to each other. They are the homologous chromosome. Here they are not identical. Here I couldn't draw them as identical, right? But actually they are identical to each other, right? They are homologous chromosomes, right? Mother and father chromosomes are the homologous chromosome of a specific type. Now this is one specific type. This is another specific type. Why is this specific type? Because this controls some features like hair color, eye color. This chromosome pair will control another set of features like the skin color, like the height, right? This pair of chromosomes will control the intelligence, right? And some other features, right? The last pair of chromosomes will determine the sex, right? Likewise, different pairs of chromosomes are for different functions, right? I hope you understood what homologous chromosomes are, right? So in actually in a cell, as I've told you, they have chromosomes like this, right? In a cell they have chromosomes like this. Now we are looking at prophase, that is after replication only in prophase happens. So this is how the chromosomes looks like. What happens is the two homologous chromosome, we have two homologous chromosome, one from the mother, one from the father. They sort of form an interaction with each other. Interaction combined with proteins, right? Look, the formation of zipper-like structure called the synaptonimal complex by specific proteins holds the two homologous chromosomes together. Okay, so the synaptonimal complex is a zipper-like structure which holds the two homologous chromosomes tightly together by specific proteins. This is the real structure. That real structure means the hypothesized structure, right? You can see the two chromosomes. This whole structure is the synaptonimal complex, right? Synaptonimal complex, this whole structure. You can see the two chromosomes. Here they haven't shown the replicated one, right? Here they have just drawn two blocks, right? To show the chromosome, right? You can see that they are attached to each other by proteins, right? That is called the synaptonimal complex, and you call it a zipper-like structure because you can see sort of like a zipper-like structure, right? Forming bridges between the two chromosomes, right? So write down, if you have, just to be interactive only, I'm giving you these questions. If you have what do you call, a full scap paper near you, or for the 2026 official students, you can take the writing book and write this down, right? What is synaptonimal complex? Or no need to write the question. If you want, write down synaptonimal complex, synaptonimal complex, synaptonimal complex, synaptonimal complex. Synaptonimal complex is a zipper-like structure. Synaptonimal complex is a zipper-like structure. Synaptonimal complex is a zipper-like structure which holds two homologous chromosomes. synaptonymal complex is a zipper-like structure which holds two homologous chromosomes, which holds two homologous chromosomes tightly together by specific proteins. Right? Synaptonymal complex is a zipper-like structure which holds the two homologous chromosomes tightly together by specific proteins, right? Which holds the two homologous chromosomes tightly together by specific proteins. Right. Now, that is the synaptonymal complex, right? That is what you call as a synaptonymal complex. What is this process called? This combination process. This pairing, you can see there is a pairing, right? Between the two homologous chromosomes. You can say that there is an interaction when the synaptonymal complex is formed like this. What is this process called as? This process is called synapsis, right? This process is called synapsis. They have given the definition as the pairing and physical connection of homologous chromosomes is called synapsis, right? You can see the synapsis. Yeah, if you can see, you can't see the synapses. I mean, you can see the synapses. This is basically the forming of the synaptonymal complex is what you call as the synapses. But the definition now, by that particular definition in your orbit, it'll be hard for you to understand if you are a 2026 student, right? So I have taken this definition. It is the connecting of two homologous chromosomes to form a tetrad. Now you can see the two homologous chromosomes are like this, right? They are paired like this. Can you see? They are paired. Here also they are paired. Here also they are paired. Now they look like a tetrad. Can you see? A tetrad. 4 chromosomes are present. Now it looks like a tetrad. This process is what you called as synapsis, right? The pairing and physical connection between two homologous chromosomes is what you call as synapsis. Basically, the formation of synaptonymal complex is what you call as synapsis. But you can't write that for the definition if they ask you. This is the definition that you need to write down if they ask it for a SCQ. Right. Now, you know what this synaptonymal complex is, right? What is synapsis? You know these things, right? Now, let's see, there is an important word called crossing over, right? Important word called crossing over. This happens due to synapsis, right? Due to synapsis only this happens. Let's see what this crossing over is, right? During synapsis, part of the DNA molecule of non-sister chromatids paired homologous chromosome break, exchange, and rejoin at corresponding point. Now it's something like this. This is the chromosome from the mother. This is the chromosome from the father, right? Mother and the father. Now they form a synaptonymal complex, right? They form the synaptonymal complex, right? You can see here they form the synaptonymal complex. This process is called synapsis, right? Synapsis, while you can see a tetrad form. Then what happens during synapsis is this particular part of the DNA present in this chromosome breaks. This particular DNA present in this chromosome breaks. And this particular DNA in this chromosome also breaks, right? Either this particular chromosome break or either this particular chromosome break, right? I mean, both break actually. This particular chromosome break as well as this particular chromosome also break, right? And what they do is they exchange these two DNA parts, right? So something like this happens. Something like this happens, right? This part will now have that black DNA. That means this father molecule will have a mother DNA molecule, and the mother chromosome will have a father DNA molecule like this, right? This process is what you call as crossing over, right? This process is what you call as crossing over. And here they are saying that this crossing over happens between non-sister chromatids. What are sister chromatids and what are non-sister chromatids? Okay, if I draw the chromosome like this, these two are sister chromatids, right? These two are sister chromatids. Again, these two are also sister chromatids. But if I draw two chromatids like this and this are not sister chromatids, right? This and this are also not sister chromatids. They are non-sister chromatids, therefore, right? They are non-sister chromatids, and the crossing over takes place between the two non-sister chromatids of two homologous chromosomes, right? Of two homologous chromosomes only, the crossing over takes place, right? So you can see the crossing over, they have clearly shown it over here. Yeah, they have clearly shown it over here. You can see the parts are exchanged. The dark purple part has come to the light purple part. The light purple part has come to the dark purple part. Again, over here also, the light purple part is in the dark purple part. The dark purple part is in the light purple part, right? You can see the crossing over afterwards clearly over here also, right? You can see the result of the crossing over, right? After that, right? You can see the father chromosome have structures from the mother chromosome. The mother chromosome has structures from the father chromosome. That is due to crossing over, right? So during synapses, part of the DNA molecule of non-sister chromatids, right? Of non-sister chromatids of paired homologous chromosomes, right? Of paired homologous chromosomes, right? Even I have made that amendment in my resource book. You can also make that amendment of non-sister chromatids of paired homologous chromosomes, right? Of paired homologous chromosomes. That means, like I showed you before, these two are non-sister chromatids, right? These two are non-sister chromatids. But non-sister chromatids are paired homologous chromosomes, right? Paired homologous chromosomes, because that they two are homologous to each other. Homologous means they two are the same. One is from the mother, one is from the father, but they both have the similar structure because they code for the same functions. Okay? All right. Now, during synapses, part of the DNA molecule of non-sister chromatid paired non-sister chromatids of paired homologous chromosomes break, exchange, and rejoin at the corresponding point. What do you call this process? This process is what you call as crossing over, right? The points of crossing over becomes visible as chiasmata. Singular is chiasmata, plural is chiasma, right? These points of crossing over becomes visible as chiasmata after the synaptonymal complex dissembles and the homologous chromosomes slightly apart from each other, right? This particular thing is what you call as the chiasmata. Chiasmata, right? Chiasmata, right? So you can see the chiasmata visible. That means you can clearly see the chiasmata visible after they have after the two homologous chromosomes have separated, before they were joined, right? You can say they were joined with the synaptonymal complex. What is that process called? That process was called as synapses, right? During synapses, crossing over took place. As a result, the chiasmata was formed, right? The chiasmata, right? This is the chiasmata formed. It will be visible. Can you see? It will be clearly visible after the two chromosomes, after the two homologous chromosomes are separated, the chiasmata is visible, right? The chiasmata is formed during the crossing over process. After that, you can clearly see that, right? Okay. Now, these points of crossing over becomes visible as chiasmata after the synaptonymal complex dissembles and the homologous chromosomes slightly apart from each other, right? So basically, the synaptonymal complex forms, crossing over takes place, and again the synaptonymal complex dissembles, right? After that, what happens? I told you the two homologous chromosomes were connected to each other in the synaptonymal complex, right? After the synaptonymal complex dissembles, they again become separated from each other. But remember, they are homologous chromosomes, right? But now they are sort of different from each other. Why? Because they have undergone crossing over, right? They have exchanged part of their genes with each other, right? But still they are homologous chromosomes. Why? Because they code for the same functions, right? If you take this mother chromosome, if you take the father chromosome, if you take this particular gene in this mother chromosome, and if you take this particular gene in the father chromosome, both of them code for the same function. For example, both of them will code for the hair color, for instance. Let's take like that, right? Both the genes code for hair color. So that's why we call them as homologous chromosomes, right? Now, can you all remember the processes which takes place in the particular order? First, chromosomes condense. Second, nucleolus disappears. Next, the synaptonimal complex forms. This synaptonimal complex is what you call as the synapses. Formation of the synaptonimal complex is what you call as the synapses. In synapses, crossing over takes place. After crossing over, chiasmata is visible, right? And then what happens? Can you all remember? In prometaphyse, only the nuclear envelope was broken, right? But in here, in prophase 1, nuclear envelope breaks in prophase 1 itself, right? Nuclear envelope breaks, right? After all these things has happened, nuclear envelope will break, right? And then centrosome will move towards the opposite poles, forming spindle in animal cells. Okay, we learned in G2 phase, the centrosomes were duplicated, right? Centrosomes were duplicated. I told you centrosomes are actually, now centrosomes, if you all can remember, centrosomes are actually some places, right? Centrosomes are places. As I've told you before, there are some places where the centrioles are located in, right? Centrioles, right? If you all can remember, the place where centriole, centriole is a cylindrically arranged structure like this, right? They are paired structures. This particular location is what you call as the centrosome, right? Centrosome. So don't think centrosome is a structure, right? What happens in G2 phase is this centrosome is duplicated. Remember, here we do not use the word replicate. Here we use the word duplicate because replication is a process which is unique to DNA, right? Here centrosome duplicates, right? That means two centrosomes are produced. That means two regions like this are produced. One centrosome goes to one pole. If the two centrosomes were here, I told you the centrosomes duplicate. And remember, the centrosomes are close to the nucleus always, right? During the prophase, after this nuclear envelope fragments, one centrosome moves towards one pole, the other centrosome moves towards the other pole, right? So what does the centrosome contains? Centrosome contains the centriole. What is the function of the centriole? Now we did cellular organelles. In even 2025 students might have done these cellular organelles in unit 2, right? What is the function of these centrioles? They form the aster and the spindle, right? So when one centrosome is here, I told you the centrioles are inside the centrosome. Inside the centrosome means centrosome is the region or the place where the centrioles are. The two centrioles are perpendicular to each other. And what is the function of the centriole? Forming the spindle. Spindle is a cage-like structure which is formed, and they form this aster towards the opposite direction of the spindle. Why? Because they need to withhold this spindle, right? Otherwise, this spindle won't have any support, right? The cage needs to be whole. So there's an aster produced as well. This part is the aster. This part is the spindle, right? So centrosome moves towards the opposite poles, forming spindles in animal cells, right? They form spindle in the animal cells, right? Why is it in animal cells only? Because, can you remember, I told you centrosomes are only present in animals. What are the structures present only in animals? Lysosomes and centrosomes, right? There are some specific structures which I mentioned before, but the easiest way to remember the two somes, centrosomes and lysosomes, they are present only in animals, right? So because of this, the spindle is only formed in animal cells, right? But spindle is also spindle is not, I'm sorry, spindle is not only formed in animal cells, it's also formed in plant cells as well. But spindle in animal cells is formed from the centrosome. Spindle in plant cell is formed from fragments of microtubules, right? Just microtubules joined together and they somehow form their spindle, right? But here, centrosome is the structure which forms the spindle in the animal cell. Okay, right. So centrosome moves towards the opposite poles. While the centrosomes are moving towards the opposite poles, they form the spindle. One centrosome forms one part of the spindle, the other centrosome forms the other half of the spindle. Finally, when they are in the opposite poles, the two halves connect like this to form the whole spindle, right? That's how the spindle is formed, right? The kinetic core of each homologue attached to microtubule from one pole or the other. Right, so basically, this kinetocore, what is the kinetocore? We learned under mitosis, the special protein, right? In the special protein, basically, which holds the two sister chromatids together is what you called as the kinetocore near the centromere, right? These what do you call these two? This special protein which holds the two sister chromatids at the centromere is what you called as the kinetocore, right? So this kinetocore region, now this is the kinetocore region, it is close to the centromere. Centromere is this circular region, right? It is close to the centromere. These proteins are close to the centromere. The kinetocore of these homologous chromosomes. That means these kinetocores of the homologous chromosomes, they attach to the microtubule from one pole. Homologous attached to other pole, while the other homologous chromosomes attached to the opposite pole, right? Now listen to me carefully. That means now let's say now there are 8 pairs of chromosomes over here, right? There are 8 pairs of chromosomes over here, right? 8 pairs of chromosomes. That means in each pair, one chromosome gets attached to the north pole, the other chromosome get attached to the south pole. One chromosome get attached to the north pole, again the other chromosome get attached to the south pole, to the microtubules from the south pole, to the microtubules from the north pole, right? Because you know the centromeres produce microtubules, right? These are the microtubules. The microtubules join and they form the spindle, right? So basically chromosomes, one homologous chromosome, if one homologous chromosome will join to one pole, while the homologous chromosome of that particular chromosome will join to the other pole, right? That's how it is meant to be happen. So it will happen in that way, right? That means one will go to one pole, the other will go to other pole. It happens randomly, right? It doesn't mean the mother chromosome should always go to the north pole. The father chromosome should always get attached to the mitochondria from the south pole. There is no such thing like that. This happens randomly, and that is why we call it as independent assortment. No one is telling anyone to move like that. No one is asking the father chromosomes to move into this particular pole. No one is asking the mother chromosomes to get attached to the microtubule from the north pole, right? No one is saying anything. This happens very randomly. That is why you call it as independent. It is independently happening, independently getting assorted into the two microtubules from the two opposite poles, right? So you call it independent assortment. This word will come under importances of meiosis. That is why I included this word, right? That is what you call as independent assortment. So what happens? Now we know that the centromeres are going to the opposite poles. Opposite poles, right? One centromere which goes to the centromere which goes to the north pole will produce microtubules like this, and the centromere which goes to the south poles will produce microtubules like that, right? And one homologous chromosome will get connected to the microtubule from the south pole, while the homologous chromosome of that particular chromosome will get attached to the north pole. Again, another chromosome will get attached to the north pole. That that chromosome's homologous chromosome will get attached to the south pole, right? Likewise, the two homologous chromosomes are distributed, right? They are distributed like that, right? So they are saying the kinetic of each homologous chromosome attached to the microtubule from one pole of the other. The homologous pair then move towards the metaphase plate, right? So basically, as I told you, one is attached to one microtubule from each pole. All of them will get arranged will reach the metaphase plate. Metaphase plate is at equal distance from the two poles, right? It is at equal distance from the two poles, so we can say it is the exact middle. So all these chromosomes reach the metaphase plate like this, right? All these chromosomes reach the metaphase plate. Each homologue will get attached to either the north pole or either the south pole, right? Likewise, only they are distributed. Okay. Now let's come into metaphase 1. Now all this time we were talking about prophase 1, right? Then what happens in metaphase 1? The pair of homologous chromosomes get arranged on the metaphase plate with one chromosome of each pair facing each pole. So they're saying the pair of two homologous chromosomes get arranged on the metaphase plate with one chromosome of each pair facing each pole. Now it's like this, right? Now you can see something like this. Right. No. Right. Now, these chromosomes are attached to the microtubules from the north pole. These are attached to the microtubules from the south pole. Now, don't worry about these north poles and south pole. Just to make sure that you get the idea of the poles only I'm using this. There are no such words like that. Okay. Just for you to get an idea of the direction only I'm using those words, right? So you can see this chromosome is homologous to this chromosome. I'm the one who's saying that. You can't see that. Okay, I'm saying this chromosome is homologous to this chromosome. How? This is the father chromosome. This is the mother chromosome, right? This chromosome is homologous to this chromosome. This chromosome is homologous to this chromosome, right? The black ones are the fathers. The red ones are the mother chromosomes, right? Black ones are the fathers. Red ones are the mother chromosomes. Right, so you can see the homologous pairs are attached from the two opposite poles, right? They are attached from the two opposite poles and all of them get arranged nicely at the metaphase plate, right? All of them arranged nicely at the metaphase plate, right? You can see all of these chromosomes get arranged nicely at the metaphase plate, right? Like this nicely at the metaphase plate. The metaphase plate is like an imaginary line, imaginary plate drawn equal distance from the two opposite poles where these chromosomes get arranged nicely right in the middle, right? This mother chromosome, father chromosome, either the mother could go up, either the father could go down. It happens independently, it happens randomly. That is why you call it independent assortment, right? So they basically get assorted in the way that they want. Somehow they arrange nicely and neatly in this particular plate called the metaphase plate, right? So that is what happens in the metaphase, right? Now, going back into the book, the pair of homologous chromosomes get arranged on the metaphase plate with one homologous chromosome of each pair facing each pole, right? You can see one pair from the homologous chromosome will face one pole, right? That means if the father place if the father chromosome of one homologous chromosome faces the north pole, the mother chromosome of that homologous chromosome pair will face the south pole, right? So one of the two will face each of the poles, right? Then they are saying both chromatids of homologous are attached to the kinetochore microtubules from one pole, and those of the other homologous are attached to the kinetochore microtubules from the opposite poles. So basically they are saying is if the father is attached, the father chromosomes, that means both the chromatids over here, right? This chromatid as well as this chromatid, the two chromatids, right? The arms are the chromatids, right? The arms of the chromosomes are the chromatids. So basically these two chromatids in this chromosome, and give me a second. Yeah, these two chromatids in these chromosomes, if they are attached by the if they are attached by a microtubule from the north pole, the two chromatids of the father chromosome will be attached by a microtubule from the south pole, right? That's what they are saying over here, right? Homologous chromosomes arranged randomly at the metaphase plane, right? This random arrangement again leads to independent assortment. No one is asking them to arrange like in this way you should arrange. No one is saying that they are getting arranged in the way they want. Therefore, it again leads to independent assortment, right? That is what you call as independent assortment, right? Now, let's move on to anaphase, right? In anaphase, what happens? Kinetochore microtubules of the spindle get shortened. Now, last week also I explained what kinetochore microtubules and what non-kinetochore microtubules are. What are kinetochore microtubules? The microtubules that are attached now. Each centromere produces a set of microtubules, right, to form the spindle, right? Some microtubules will not be attached to any chromosome, but some microtubules will be attached to the chromosomes, right? Some microtubules will be attached to the chromosome, right? Some might not be attached to the chromosome, right? Some might not be attached to the chromosome. The ones which are attached to the chromosome are what you call as kinetochore microtubules because they are attached to the kinetochore protein, right? They are attached to the kinetochore protein. That's why you call them. That's why you call them as kinetochore microtubules. The ones which are not attached to the microtubules, the ones which are not attached to the chromosomes, are called as non-kinetochore microtubules. Okay, and here I wanted to say something else as well. In mitosis, can you remember the that means only one chromosome was arranged like this. Microtubule from one side also gets attached to the microtubule. Microtubule from the other side also get attached to the same chromosome like this, right? And it breaks the two sister chromatids, and one sister chromatid is taken to one pole and the other sister chromatid was taken to the other pole. But in meiosis, what happened? In meiosis, the two homologous chromosomes were arranged, right? So to one pole, they take one homologous chromosome. To the other pole, they take the other homologous chromosome, right? So you can see a clear difference between mitosis and meiosis 1, right? Because they take the homologous chromosome only to one pole, there's a reduction taking place. Let's see. Still you won't understand, still we haven't completed it. Let's see, right? Kinetochore microtubules of the spindle get shortened. What do you mean by kinetochore microtubules of the spindle get shortened? Before they were long like this, right? The ones which were attached to the homologous chromosome, right? Ones which were attached to the homologous chromosome, right? Before they were long like this, and there are red ones as well, right? They were arranged in the metaphase plate, right? There are non-kinetochore ones as well, right, which are not attached at all. So they get shortened. When you shorten an elongated root, you have to pull it, right? You have to pull it, right? So basically, these microtubules are getting pulled towards the pole. That's why they are getting shortened. They are getting pulled towards the pole. When they are getting pulled towards the poles, what happens to these chromosomes? These two homologous chromosomes, The ones which are attached to the microtubules from the North Pole will start moving to the North Pole, while the ones which are attached to the microtubules from the South Pole will start moving towards the South Pole, right? The ones which are attached to the microtubules from the North Pole will start moving towards North, and the others will start moving towards the South, right? Okay. Homologous pair separates and one chromosome of each pair move towards the opposite pole, right? So I will take the diagram again, then it's easy to explain. You can see in this diagram, right? You can see in this diagram when these chromatids get short, when these microtubules get shortened, when these kinetochore microtubules get shortened, can you see these are the non-kinetochore microtubules? When the kinetochore microtubules get shortened, they pull these homologous chromosomes towards the poles. So the black chromosome, let's take black is father, red is mother. This father chromosome will move upward, and the homologous chromosome of that father chromosome, that means its mother chromosome, will move towards the opposite pole, right? Will move towards the south pole. You can see the two homologous chromosomes are getting separated, right? So now they are not going to stay homologous again, right? Because they are going to get separated. Before they were together, right? Before these two chromosomes were together, right? Now they are going to pull these two towards the opposite poles. So they are not going to remain closer to each other for any. That means no longer are they going to stay closer to each other, right? Homologous pair separates and one chromosome of each pair move towards the opposite pole, right? Either the black goes to the North Pole or either the red goes to the North Pole. It depends on how they were arranged in the metaphase plate. We know they were arranged randomly. So sometimes many blacks would go to the North Pole, sometimes many reds would go to the South Pole. Depends, right? It's just independent assortment, right? It happens in the way that they like, right? Sister chromatids of each chromosome remain attached at the centromere and moves as a single unit towards the same pole. Why are they saying, why are they talking about sister chromatids over here? Because in mitosis at anaphase, the sister chromatids were the ones which separated, right? The sister chromatids were basically separate. One sister chromatid moves upwards while the other sister chromatid moves downwards, right? That's how they maintain genetically identical offsprings, right? That's how they maintain genetically identical offsprings. But here they are taking one homologous chromosome to one pole, and the pair and basically the part of that pair to the other part of the cell, right? It means they are basically separating the two homologous chromosomes. That means they are basically separating the pairs. Now I have drawn the pairs before also. If they are going to separate the pairs, and at the end, two cells are formed. Now, there were 8 pairs initially, you can see, right? There were 8 pairs initially. If they are going to separate these two homologous chromosomes, and if they are going to bring it into two poles, you know, these poles will at the end form 2 cells. Then how many pairs will be inside one chromosome, inside one new cell? 4 pairs, right? Inside one new cell, you will only find 4 pairs, right? Because these were separated. The two homologous chromosomes were separated. So at the end, each new cell will have how many? 4 pairs. Can you see that there is a reduction from 8 pairs to the new cells? Now there are 4 pairs. That's why you call meiosis 1 as the reduction division, right? That's why you call meiosis 1 as the reduction division. Any questions? If you have any questions, you can ask me through WhatsApp. Okay. Right. So that's why you call it a reduction division. In mitosis, what happened? In mitosis, the two chromatids separated, right? The two chromatids separated like this. Wait, I will take a different color. The two chromatids separated. After the two chromatids separated, that means if you take, for easiness, I will take just 4 pairs of chromosome. Forget everything. Talking about mitosis now, right? Talking about mitosis. I will take 4 pairs of chromosomes are present in a particular animal. Okay, let's take 3 pairs of chromosomes are present in a particular animal. This special animal
has only 3 pairs of chromosome inside it. A special animal, okay? It has only 3 pairs of chromosomes. What happens in mitosis? Now, these are in the replicated form. That means after they are replicated, what happens in mitosis? What happens in mitosis? The sister chromatids are going to get separated towards the opposite poles, right? That means one sister chromatid, that means how many sister chromatids will go towards the north pole? 1, 2, 3, 4, 5, 6. 6 sister chromatids will go toward the north, towards the north pole. Again, how many sister chromatids will go towards the south pole? Because they are going to divide it by the sister chromatids. Again, 6 will go down, right? Again, 6 will go down, right? And you know, they were replicated because of that. And again, you can see how many pairs of chromosomes will be there at the end. Again, 3 pairs of chromosomes will be there in the new cell, right? Again, 3 pairs of chromosome will be there in the new cell. 1 pair, 2 pair, 3 pair. 1 pair, 2 pair, 3 pair, right? In mitosis, identical cells were produced. Diploid cells were produced. But in meiosis, because the homologous chromosomes, not the chromatids, because the homologous chromosomes are the ones which gets divided, only half. That means there's going to be a reduction. If there was 2n, there will be n. If there were 8 pairs, there's going to be only 4 pairs at the end of meiosis 1. Meiosis 1, right? I hope you understood that point. Do you have any doubts? If you have any doubts, you can ask me now, right? If you have any doubts, you may ask me now. If you didn't understand any part, you can ask me any questions, right? Right. Okay. Now, that was the difference between, that's how in mitosis diploid becomes diploid, and in meiosis 1, that's how diploid becomes haploid, right? These chromosome sets, everything will be learned in detail under genetics. For now, you just need the simple, simple knowledge of these chromosomes. That's it, right? Okay. Now, telophase 1. In telophase 1, what happens? Now, in anaphase, the two homologous chromosomes separated, right? They separated into the two opposite poles. In telophase, what happened in mitosis? In mitosis also, the nuclear envelope again reformed, right? The disappeared nucleolus again reformed in the telophase, right? Likewise, the ones which were disappeared before in the prophase appeared in the telophase, right? So basically, this is like the appearance process. Everything which has disappeared in the prophase starts appearing in telophase, right? So you can see in telophase, one complete haploid set of chromosomes accumulate at each pole, right? Why is it haploid? Because the chromosomes number are reduced now, right? Because one pole will have only, if there were 46, if there were 20, now if there were 8 pairs initially, in the mother chromosome, 4 pairs goes to the north pole, 4 pairs goes to the south pole, right? So you can see the number has reduced. That's why you call it one complete haploid set of chromosomes accumulate at each pole. Now at each pole, there's going to be a haploid set of chromosome, and around this haploid set of chromosome, the nuclear envelope reforms around each of these chromosomes, right? Around the chromosomes, the nuclear envelope will reform. That means the chromosomes will be incorporated into a nucleus. Now they are covered by a nucleus, right? And I told you at prophase the nucleolus disappeared. Now again, 2 nucleoli will appear. That's why they say nucleoli, plural of nucleolus, right? The nucleoli reappear. And there was a spindle formed, right? The spindle was formed between the 2 poles, right? This spindle will disintegrate. It will be broken down, right? Remember the order in telophase also. This order is important. First, haploid set of chromosomes get accumulated. Next, nuclear envelope reforms. Next, nucleoli appears. Next, spindle disintegrates. Now it happens in the way that it disappears itself. Can you remember? First, at last, what happened? At last, only the nuclear envelope
disappeared, right? At the end of prophase only the nuclear envelope disappeared. At the start of prophase only the nucleolus disappeared, right? That means at the beginning of telophase, the nuclear envelope will form. Why? At the end of prophase only disappeared, right? So first, the nuclear envelope should form for the nucleolus to appear, right? You have to remember this order. That's why I'm saying this, right? So there was an MCQ on the order in a past paper. So they can test you anytime from any part of this meiosis part, right? So basically, in prophase 1, what happens first? The nucleolus disappears first. Then only the nuclear envelope reforms. In telophase 1, first the nuclear envelope reforms, then the nucleolus reappears, right? Then the spindle will disintegrate, chromosomes decondense. Decondensed means opposite of condensed, which means becoming thinner, right? They become thinner. I told you chromatin, the chromatin, when it becomes condensed only, you call it chromosomes, right? When it becomes condensed only, you can call it chromosomes. Last week I have explained that in details, right? Right? So chromatin becoming condensed, that is what you call chromosomes. Genetically non-identical haploid 2 daughter nuclei are formed within one cell, right? You can see that means these 2 nuclei which is formed. Now when a nuclear envelope forms, these become nucleus, right? Nuclei, right? You can see they are haploid, right? And they are saying they are genetically non-identical. Why? Because the chromosomes, the pair of chromosomes randomly got distributed, right? They got randomly distributed. Now, if there were 8 pairs of chromosome, out from those 8 pairs, we know out from those 8 pairs, we know 8 pairs means 16 chromosomes. We know 8 chromosomes are mother, 8 chromosomes are from the father, right? 8 chromosomes are from the mother, 8 chromosomes are from the father, right? That means over here, what will happen is, over here, we can't say all the eight mother chromosome will go to one side and all the 8 chromosome will go to one side. We can't say like that, right? All the 8 mother chromosomes will go to one particular north pole. All the 8 chromosome will go to one particular south pole. Then we can say that they are identical, right? Then we can say, still we can't say that they're identical because of the sex chromosomes. We can't say that they're identical. Anyhow, you can understand when the 2 homologous chromosomes get distributed like this, the combination it forms over here is different from the combination it forms in the north pole. The 2 combinations are different, right? One can be a mother chromosome, one can be a father chromosome, again a mother, right? Father, right? Mother, father, likewise. The 2 are different, right? The 2 are different, right? So basically, they're non-identical, right? Non-identical. That's why in sperms, sometimes sperms will carry annex chromosomes. That's why some sperms will carry the Y chromosome, right? They are non-identical, right? All these sperms are not identical to each other, right? That means these are non-identical to each other, right? All are sperms, but they're non-identical. One can carry the X chromosome, one can carry the Y chromosome, right? Okay, right. Now, basically, that means in telophase, the 2 nuclei forms inside the cell, right? In cytokinesis, this cytoplasm divides to form 2 cells, right? This cytoplasm divides to form 2 cells, right? Cytokinesis usually occurs simultaneously with telophase 1. Cytokinesis, that is an important MCQ point. Underline that, usually occurs simultaneously with telophase 1, right? Usually occurs, that means at the same time with telophase 1 only it happens, right? But in mitosis, they have said the division of the cytoplasm starts at the end of the telophase. Now, these are good MCQ points. In mitosis, go back to your mitosis, page number 41. You can see there they have said, I will also go to that page number, right? Page number 41, the division of cytoplasm starts at the end of telophase. But here can you see cytokinesis simultaneously occur with telophase, right? Cytokinesis simultaneously occur with telophase, right? Cytokinesis simultaneously occur with telophase. Genetically non-identical haploid two daughter cells are formed, right? In animal cells, cleavage furrow is formed. In plant cells, a cell plate is formed. Right. You can see this cytokinesis takes place in animal cells by forming a sort of hole-like structure, not a hole, actually a cleavage-like structure to the inside, which you call as the cleavage furrow. Right. The cytoplasm goes to the inside. They forms like a cleavage, and then slowly this cleavage extends and these two cytoplasm get separated in the animal cells, right? In plant cells, there are Golgi apparatus. The Golgi apparatus forms Golgi vesicles. These Golgi vesicles arranged in the middle like this, right? And then they combine together, forming the cell plate. The cell plate divides the two cytoplasm, right? The two cell plates divides the two cytoplasm, right? So that's what happens in animal cells and in plant cells, right? Why can't cleavage furrow takes place in plant cell? In mitosis also I told you, the plant cell have a cell wall, right? The cell wall can't be, that means the cell wall can't undergo that cannot form the cleavage furrow, right? Therefore, the cell wall, that means to join the cell wall along, that means the cytoplasm is intact, the cell membrane is in contact with the cell wall, right? So basically we have to form this vesicle, we have to form this cell plate to divide it completely from the cell wall to form two new cells. But in animal cells, there's no cell wall, so the cleavage furrow can be formed easily, right? The cleavage furrow is formed. So that is the difference between cytokinesis, and that is the difference between, give me right. Now let's start cytomeiosis 2. Now in meiosis 1, the centromeres, if you take the orientation, right? If you take the orientation of the spindle formation, the spindle formed, if it formed vertical in the meiosis 1 phase, in meiosis 2, the spindle will form perpendicular to the first orientation. In meiosis 2 also there will be a spindle forming. In meiosis 2, the spindle form will be perpendicular. The orientation will be perpendicular. In meiosis 1, if it was horizontally formed, then in meiosis 2, the orientation will be vertical. Likewise, the orientation will be perpendicular from meiosis 1 to meiosis 2, right? Depending on what the orientation is at meiosis 1, right? Okay. Now meiosis 2, you call it meiosis 2 also, we know that the subphases are there: prophase, metaphase, right, anaphase, and the telophase, right? Let's see now. See here in the cytokinesis, they say no DNA replication occurs. Why are they saying this? Because we are actually taught to understand. That means if we just memorize, we would think, okay, before prophase DNA replication takes place, before prophase happens, we'll always think, okay, interphase needs to take place, right? We'll think like that. But no, before prophase 2, that means before meiosis 2, no DNA replication takes place. That means there is no interphase again. In meiosis, only one interphase is present. That is at the very beginning, right? After that, meiosis 1 takes place. After that, meiosis 2 takes place. In between, no interphase present. That means no DNA replication will occur. Okay, right. Then let's see what happens in prophase 2. Why do we call it? It's just the numericals, right? We can't give them, we can't call the prophase a different name, right? If the same thing is happening in both the phases, it's easy for us to give the same name, but just to tell us that they are in two different stages only, we use the two numericals, right? So prophase 2 is to show that this prophase takes place in meiosis 2, and you need to be careful when you write these things, right? Sometimes you might just write prophase, which is wrong. So always make sure you write you have to specifically indicate which prophase it is when it comes to meiosis, right? In prophase 2, centrosomes start producing spindle apparatus. Right, now in meiosis 2, at the end of meiosis 1, 2 cells were produced, right? 2 cells were produced. Each of these will undergo meiosis again, right? Will undergo meiosis 2. Each of these cells will undergo meiosis 2, right? Each of these cells will undergo meiosis 2. So the centrosomes present will start producing the spindle apparatus, right? If they ask you what this spindle apparatus is, basically spindle is like the cage-like structure. If you take this whole apparatus, what does it include? It includes the spindle fibers. Okay, that means the microtubule fibers, right? The microtubules, the spindle fibers, the aster, right? Aster, you know what is the aster, right? The aster is like the holding structure. It is also made up of microtubules. And this centrosome. What is the centrosome? The place where the pair of centrioles are located is what you called as the centrosome, right? So the spindle apparatus has spindle fibers. It has an aster. It has a centrosome, right? So you can see centrosomes start producing spindle apparatus. In the previous one also, basically we saw the spindle was formed during the prophase 1, right? In all the phases, in mitosis, in meiosis 1, in meiosis 2, the spindle is the spindle actually started getting formed at prophase, right? It started forming at the prophase, right? Chromatin fibers condense. Okay, that means I told you when chromatin gets condensed only, that means basically the mesh-like structure I told you is called the chromatin. When it gets condensed only, you can see the beautiful structure, right? You can see the beautiful structure of the chromosome. Actually, you see it like this because they are replicated, right? You see it like this because they are replicated, right? So chromatin, when they condenses, they become chromosome. Condenses means when they become thicker, you call it chromosome, right? Chromatin fibers condense and produce chromosomes with 2 sister chromatids. You know, they look like 2 sister chromatids, right? 2 sister chromatids are there. This is one sister chromatid. This is the other sister chromatid, right? That's how the chromosomes look like, right? After replication, okay? After replication, that means here no replication takes place, but initially replication took place, right? At the start of meiosis itself, replication took place in the interphase, right? Because of that, the chromosomes appear with 2 sister chromatids like that. And again in prophase, nuclear envelope breaks down into fragments. Nucleolus disappears. look at this. Here they have given a different order. In prophase 1, first the nucleolus disappears, then the nuclear envelope broke down, right? Broken down, right? But here, first the nuclear envelope breaks down, then the nucleolus disappears. Sometimes it's confusing, so these minor details needs to be studied closer to an exam. That means when you are coming for my paper classes in the future, you have to study these parts right before each of the papers that you are sitting for. That's how I have trained the 2024 students as well. I have given them a specific list which they need to be studied before coming for all the papers, right? So you also will have to study. That means it's a must. It depends on you whether you do it or not, right? So you also need to study the minor details, right? Usually for 2024 students, I didn't give these minor details because they joined the class late in the August only I started, right? So I have given them some tables to be studied and everything. For y'all, I will give these minor details as well, right? So these minor details are to be studied at the close of the exam, like the orders and everything closer to the exam. But the processes and everything needs to be studied tomorrow itself. That means tomorrow when you have time, you have to study this part. Then during the week, you have to revise this part. Next week also, you need to do a revision on this part, right? That's how you can put these things into your long-term memory, right? Afterwards, when the weekly bundle comes, you can attempt the weekly bundle questions as well, where you will recall all of them, right? Okay. Now, you can see during the late prophase 2, that means basically at the end of the prophase 2, centromere of the chromosomes are moved to the metaphase plate. Centromere of the chromosome are moved to the metaphase 2 plate. So basically, the chromosomes get arranged at the metaphase plate. The centromeres get arranged. At the end, the centromeres get arranged at the metaphase plate in prophase 2, right? That means now I told you there are 2 cells produced at the end of meiosis 1, right? That means if there were 8 pairs initially in the mother chromosome, now 4 pairs are only there. 4 pairs are only there in each of these cells, right? And those 4 pairs of chromosomes, 4 pairs means 8 chromosomes, right? The 8 chromosomes will get arranged in the metaphase plate, right? 1, 2, 3, 4, 5, 6, 7, 8. 8 chromosomes will get arranged in the metaphase plate like this. The centromeres will be located in the metaphase plate, right? There's going to be 8 chromosomes located in the metaphase plate because there are 4 pairs of chromosomes, right? Okay, now let's see what happens at the metaphase. At metaphase 2, all chromosomes get attached to the microtubules at their centromere. So all these chromosomes get attached to microtubules from the North Pole as well as the South Pole like this, right? These chromosomes get attached to the microtubules from the North and the South Poles, right? At their centromere. Centromere is that circular sort of region, right? In the chromosome, right? Which attach the 2 sister chromatids together, right? And aligned on the metaphase plate. You can see they are aligned on the metaphase plate, right? They get aligned on the metaphase plate. Can you see? Here the orientation in meiosis 1. Here the orientation in meiosis 1 was vertical. But can you all see here the orientation of the spindle formation is horizontal? Can you all see the orientation is perpendicular to one another in meiosis 1 and meiosis 2? That is an important thing to be noted down, right? So you can see the chromosomes are actually held together held at the metaphase plate, right? The chromosomes are held at the metaphase plate, right? You can see one microtubule from each pole will attach to the chromosome. One microtubule from each pole will get attached to the chromosome. But here, can you see? Here, can you see? Even though these are 2 homologous chromosomes, right? This chromosome wasn't attached to a microtubule from the opposite pole, right? The homologous pair of the microtubule was attached to the opposite pole. But here, the same chromosome is attached from the North Pole microtubule as well as from the South Pole microtubule, right? So when they start pulling towards the opposite poles, what happens is at the centromere, it gets broken down because one pole pulls it upwards, the other pole pulls it back, pulls it downwards. So at the centromere, it breaks down. Therefore, the sister chromatids get separated, right? That is the difference between meiosis 1 and meiosis 2. So you can see kinetochores of sister chromatids are attached to microtubules extending from both the poles. Like I said, the kinetochore, what is the kinetochore? The protein which attach the two sister chromatids together at the centromere is what you call as kinetochore, right? Now there is a there was another protein called cohesin in mitosis. Don't confuse it with cohesin. I have explained cohesin in mitosis, right? Kinetochore is the kinetochore is the protein which attaches the two sister chromatids at the centromere, right? It is a protein, right? So this kinetochore over here is attached to one pole or from the one side, as well as this kinetochore is attached to one pole from the other side. So you can see the same chromosome is attached from both the sides, from both the poles, right? Microtubules extending from both the poles. That is the importance over here, right? Due to crossing over in meiosis 1, the two sister chromatids of each chromosome are not genetically identical. Now, if you take this chromosome, due to crossing over, one chromosome will look, the chromosome will actually look like this, right? So are these two sister chromatids, is this chromatid equal to this chromatid? These are sister chromatids, right? Are these sister chromatids identical to each other? No, they're not identical. You can see there is a crossing over taken place over here. So you can't say that they're identical. That's why they become genetically non-identical in meiosis 2 also. Right? In meiosis 1 also they become genetically identical. In meiosis 2 also they become genetically identical due to this crossing over. Right? So basically, due to crossing over in meiosis, the two sister chromatids of each chromosome are not genetically identical. Meiosis 2 usually takes place in the perpendicular direction of meiosis 1. Therefore, metaphase plate of meiosis 2, underline, this is a good MCQ point. Therefore, metaphase plate of meiosis 2 is perpendicular to the metaphase plate of meiosis 1. Right? Metaphase plate of meiosis 1 is perpendicular to the metaphase plate of meiosis 2. If the metaphase plate formed in meiosis 1 is horizontal, the metaphase plate formed in meiosis 2 is going to be vertical like that, right? Okay, now, yeah. Okay, now let's move on to anaphase 2, right? I was planning to ask the true false questions, but I'll ask that at the end. Anaphase 2, right? At anaphase 2, due to break, now they're saying anaphase 2, what happens? The microtubules, usually the microtubules get shortened and they pull the two sister chromatids away from each other, right? They pull these two sister chromatids away from each other. So they break down at their centromere, right? You see it is same as mitosis. Meiosis 2, basically there are a lot of similarities, right? A lot of similarities, but there are some differences as well. Why? Because there's no, that means that the in mitosis genetically identical, the sister chromatids are genetically identical, but in meiosis 2 they are not genetically identical, right? They are not genetically identical in meiosis 2, right? So they are not exactly same. That's why you use the word similar, because basically the process is similar, right? In mitosis as well as in meiosis 2. Due to breakdown of proteins attached in sister chromatids, what are the proteins? Kinetochore, and what is the other protein? The cohesin, right? The other protein is cohesin, and kinetochore, the sister chromatids are attached using this, they get broken down, right? They get broken down. Now this is the protein which attach the two arms of the sister chromatids, is what you call as cohesin. The protein which attach the two sister chromatids like this is what you call as kinetochore, right? So when these proteins get broken down, one part can be pulled up while the other part can be pulled down, right? Due to the breakdown of proteins attached in sister chromatids, They are separated at the centromere. As a result of shortening of microtubules, sister chromatids of each chromosome move towards the opposite pole, right? Sister chromatids of each pole, that means this sister chromatid will move towards this pole, and this sister chromatid will start moving towards this opposite pole, right? So that's what's going to happen under anaphase 2, right? Then in telophase, now you know in telophase, basically everything that has disappeared will start reforming again, right? So you can see nuclear envelope reforms, nucleolus reforms, and you can see the thickened chromosomes become thin again. When they become thin, you call it chromatin. Chromatin condenses into chromosomes condense into chromatin. The spindle will disassemble, right? And at the end there will be genetically, that means now I have drawn now at the end of give me a second, yeah, okay, right. So now in now due to meiosis 1, 2 cells were produced, right? Now this was initially there were 8 pairs of chromosome, now 4 pairs over here, 4 pairs over here, right? Now it undergoes, each of these cells undergo meiosis 2, right? Each of these undergoes meiosis 2, and again it produces 2 cells, right? You know, after each cell division, 2 cells are produced. Cell dividing means producing 2 cells, right? Dividing to form 2 cells, right? So here again 4 pairs are there, 4 pairs are there, 4 pairs are there, 4 pairs are there, because meiosis 2 is similar to mitosis. The sister chromatids are getting separated, so there is no reduction in the chromosome. But here, due to the separation of the homologous chromosome, there will be a reduction in the chromosome number. That's why 8 pairs become 4 pairs. But in meiosis 2, 4 pairs are still remain, right? And you can see 4 cells are produced at the end of meiosis process. Whole meiosis process, 4 cells are produced, right? So you can see the clear difference between the mitosis and meiosis, right? Meiosis as a whole, right? As a whole means including meiosis 1 as well as meiosis 2. So at the end of telophase 2, genetically non-identical haploid 4 daughter nuclei are formed from one parent cell, right? From one parent cell, that means if you take, this is the one parent cell, right? One parent cell, if you take one parent cell at the end, you can see at the end of meiosis 2, you can see 4 genetically non-identical haploid daughter nuclei. After cytokinesis only, they become 4 genetically non-identical haploid daughter cells, right? So you can say cytokinesis occurs as in mitosis. As in mitosis means what? In mitosis, cytokinesis happened after telophase, right? Happened at the end of telophase, not simultaneously with telophase, right? But in meiosis 1, cytokinesis happened simultaneously with telophase, right? But here it occurs as in mitosis. That means what? It occurs at the end of telophase. Genetically non-identical haploid 4 daughter cells are produced. These 4 daughter cells are not even identical to their parent cell, right? That means they are identical, they are non-identical to each other, right? And also they are non-identical to their parent as well, right? They are non-identical to their parents. So if this is 8 pairs, you can see at the end 4 pairs are produced. This is how our gametes are produced. What are our gametes? Meiosis is involved. Now, can we produce our normal cells? If we produce our normal cells with meiosis, all our cells will be haploid. Then we can't do our functions because haploid cannot actually exist. That means haploid skin cells, there are no such thing like that. We can't do our functions if our cells are going to be haploid. But gametes are haploid. Why? Because then they can combine. Sperm and egg can combine and form a diploid cell at the end, right? Sperm and the egg, which are haploid, can combine and form a diploid cell at the end. So all these are in meiosis is involved in the formation of gametes. Gametes are what? The eggs in females, the sperms in the males, right? So the egg and the sperm, which are haploid is indicated by N, they combine and form diploid again, right? Form the human or the animal or the plant, right? Which is diploid again, right? So actually, these diagrams are needed. I will explain the diagrams as well. Now, if you get an essay you got an essay, actually 2022, you got an essay on meiosis 1, right? Let's go to that part, 2022. Yes, 2022, look at the essay that they have asked. Describe the nuclear division. Describe the nuclear division where the chromosome number is reduced. What is the nuclear division where the chromosome number is reduced during meiosis? That is meiosis 1. Now, if you were a student who thought they have asked without reading the question properly, you think you had to write an essay on meiosis, and you write both meiosis 1 and meiosis 2. First of all, they would think sometimes you can lose marks because they would think that you do not know what is the chromosomal number, what is the nuclear division where the chromosomal number is reduced. So by thinking like that, they can cut your marks, right? But if they are very generous and if they don't want to cut their mark, if they don't want to cut your marks, they will mark the meiosis 1 part and give you marks, and they will ignore the meiosis 2 part depending on the examiner, right? If you have written both, they will ignore. But still there is a disadvantage for you. Why? Because you have spent a lot of time in writing meiosis 2, which you could have spent for another essay, right? So your time management gets completely destroyed at the exam. That's why you have to take 10 minutes to read the essays carefully, right? You have to take 10 minutes. That's why you get 10 minutes reading time to read the essays carefully and choose the best essay that you can write, right? And you can see they have given all the points. This will be a homework for all the students. Without looking at the marking scheme, you have to write that. After that, you have to check with the marking scheme, right? After that, you have to check with the 2022 marking scheme. If you were a student who thought, okay, the diagrams, usually the diagrams play a major role. Sometimes the diagrams are given 5 marks, and those 5 marks are also important for us, right? But sometimes if you are if you are a smart student, smart does not mean intelligent. Smart means you're tricky. If you are smart, that means you won't draw these diagrams at your exam. You know, these diagrams, you should study these diagrams. That means you should practice these diagrams at home. In case you get an MCQ, a structured essay to draw, right? You should practice these things. But for an essay, don't. That means if you spend some time, that means if you spend time to draw these structures, see, you get only 3 marks from 150 marks. This 150 marks is finally converted to 7.5 marks. Will this 3 marks have any effect? Will it have any single effect on your final mark? No, not at all. So if you spend around like 20 minutes drawing this diagram perfectly, that's a waste of time. So first of all, we got chloroplast we got photosystem too. First of all, I drew a quick sketch, right? Afterwards, I thought after I finish, you have to draw diagrams in some important scenarios. You get 5 marks. So for photosynthesis, I quickly drew the 2 photosystems when it came to my essay, and I quickly wrote down all the points. I finished everything. I drew the sketch and I labeled as well quickly, but I didn't draw them in detail. At the end, after I finished all the essays only, I again came and completed all the diagrams fully, right? I mean, I drew the diagram, but if I have ever got this meiosis to be drawn, I wouldn't ever draw even a sketch. I will just skip that part, just write the points down, keep some space to draw it later if I have some time at the end, right? If I've got some time at the end, I will draw this part, and then that's how I'll do that, right? So you have to be a bit tricky, right? This is a homework question for all the students. After that, you have to look at the marking scheme and mark your points as well. First, you need to study meiosis 1, then you need to write, then you need to, I will send the homework separately, the time duration and everything to the group, right? So you can do that later, right? Okay. All right. Now we have to complete the remaining part. Right. We have a small part. We have to talk about the diagrams, right? Okay. Now talking about the diagrams, you can see this is meiosis 1. First we are talking about prophase 1, right? You can see in meiosis 1, here they have only taken 3 pairs of chromosome, an animal with 3 pairs of chromosome. These two are 2 homologous chromosomes, right? These two are 2 homologous, these two are 2 homologous, these two are 2 homologous. You can see in prophase 1, the synaptonymal complexes are formed like this, right? And there, here you can see the crosses. These are chiasmata, right? You can see the spindle has started to form, but no, not a complete one. Still the spindle is forming, and you can see the small aster like this. Here, can you see the 2 centrioles perpendicular to each other? One centriole is like this, one centriole is like this. They are perpendicular to each other. This region is what you call as the centrosome, right? Centrosome is the region with the centriole pair, right? And you can see the nuclear envelope gets fragmented and the nucleolus has disappeared, right? That's what happens in prophase 1. In metaphase 1, you can see the spindle has formed beautifully, right? There are non-kinetochore microtubules, and the 2 homologous chromosomes are attached. They are randomly arranged at the metaphase plate, right? You can see this imaginary line, just like the equator in our earth. Imaginary line called the metaphase plate is present, right? And centromere with the kinetochore, centromere is this structure, right? This structure which holds the 2 sister chromatids, that is the microtubule. You can see the microtubules are attached to the kinetochore, right? And here you can see the anaphase 1. In anaphase 1, the microtubules get shortened, right? The microtubules get shortened and the 2 homologous chromosomes are pulled. Can you see the 2 homologous chromosomes are different now because of the crossing over? That's why we say 2 genetically non-identical daughter nuclei are produced at the end of meiosis 1 also, right? Can you see these are actually non-identical to each other, right? These chromosomes are non-identical to these chromosomes, right? That's why 2 genetically non-identical chromosomes are produced at telophase, and cytokinesis takes place together at meiosis 1. So you can see basically the cleavage furrow is formed, right? Then in meiosis 2, right? In meiosis 2, in meiosis 2, here they have given 2 cells because at the end 2 cells are produced. Each of these cells will undergo prophase 1 as well as prophase 2 as well as metaphase 2. You can see initially the spindle was formed vertically, but now the spindle is formed horizontally, right? The spindle was formed horizontally, right? And you can see over here, even though here they have sent telophase 2 and cytokinesis here, even though they have given it as one diagram, you know, at the end of telophase only cytokinesis takes place in meiosis 2, because it happens just like in mitosis. They have mentioned that, right? You can say the sister chromatids are the ones which separate here, not the homologous chromosomes. Therefore, there's no reduction taking place. Therefore, still it will be the same chromosome pairs, right? Still it will be 4 pairs in each, right? Now we are coming to significances of meiosis, right? Significances of meiosis. Let's see. Maintains the constant number of chromosomes through generations in sexually reproducing species. Right. So what do you mean by this? Now, we know in sexual reproducing species, gametes are formed, right? N and N gametes are formed, and they only combine to form 2N, right? So you can see the 2N is maintained in all the generations, right? Without getting, actually, that means the 2N is maintained in all the, in all the generations. What if the gametes were formed from mitosis? If the gametes were formed from mitosis, then it will be 2N, 2N, right? The gametes, if they were formed from mitosis, it will be 2N, 2N. That means in each generation, the diploid number will change. That means our next generation will have 4N. The next generation after that generation will have 8N. If the gametes are going to be formed using mitosis, right? And in molecular biology, you will learn that the only viable set of ploidy level for humans is only diploid level, right? Only viable ploidy level, right? That is diploid level. If we have 4 and 3, of if we have 3, if we have 4 and 8 and 5 and 6 and 7 and like that, we can't survive, right? We can't survive, right? We are formed in a particular way, right? Humans won't survive, then a new species will evolve, right? If it happens like that, a new species will evolve, right? Okay, so that is one significance. It maintains constant number of chromosomes. Second one is produce new genetic variations leading to evolution. Now, due to crossing over, you can see there is a variation, right? In mitosis, there was no variation. Everyone was equal to one another. Everyone was identical. But in meiosis, you can see that there is a variation in the cells, right? When variation is present only, it leads to evolution. You will learn that in Unit 3. When you have variation in a species, some species can actually survive in a particular environment. Some species cannot survive. Those which survive can get evolved sometimes, and they can actually and that's how actually evolution takes place, right? So basically, when you produce new genetic variations, it can lead to evolution, right? Sometimes there is a thing like this. Now we are not using our toes that much, right? So some scientists are predicting that in the future our foot will not have any toes. Our foot will look like this, right? That is due to evolution, right? Because we are not using the toes, right? Some scientists are saying like that, right? That is due to genetic variation. Variation takes place due to crossing over, right? Likewise, they will due to some variations which takes place, we won't have some certain features, or we will gain some certain features, right? It leads to evolution. And you can see genetic variation occurs due to crossing over. You can see the variation in mitosis. No crossing over takes place, therefore no variation. But here crossing over takes place. And after crossing over takes place, they recombine together, right? That means the two pairs of genes recombine with each other, right? If you don't know the definition of recombination, you can write down this definition, right? Recombination is basically like this, okay? Now, if you take the two chromosomes, they form the synaptonymal complex like this. This DNA breaks down and they exchange. That exchanging process is what you call as recombination. You can write down recombination, near recombination in your resource book, write down. When two molecules of DNA, when two molecules of DNA exchange pieces of genetic materials, when two molecules of DNA exchange pieces of genetic material with each other. That is what you call as recombination. When two molecules of DNA exchange pieces of genetic materials with each other. When two molecules of DNA exchange pieces of their genetic materials with each other. When two molecules of DNA exchange pieces of genetic materials with each other. When two molecules of DNA exchange pieces of their genetic material with each other. Right. Now, right after that, you can see there is independent assortment. If you want, if any one of you missed, ask me, I will send this. I will tell that again. Independent assortment, right? You can see independent assortment. Near independent assortment, write this down. The random orientation, write it now itself. Don't try to watch the recording and write it down. It's a waste of time, even for 2026 students, right? The random of, you can watch the recording after that just to revise, but not to write these things. Write it now itself. The random orientation of homologous chromosome pairs. I will write it down. The random orientation, this is independent assortment. The random orientation of homologous chromosome pairs during metaphase 1, during metaphase 1 is what you call independent assortment, right? The random orientation of homologous chromosome pairs during metaphase 1. What do you mean by that? That means you know the homologous pairs, the father, the mother, the mother, the father, they arrange randomly at the metaphase plate. No one is saying this father should go to the North Pole, this mother should go to the South Pole, this father should go to the North Pole. No one is saying like that, right? They arrange randomly. That's what you call as independent assortment. So how is genetic variation produced? It happens due to three things. This was asked. No, this wasn't. Oh, this was. No, I think this wasn't asked. Crossing over recombination. This was asked in a model paper. Crossing over This was asked in 2020 Mora paper, right? Mora paper. It was asked. Crossing over, recombination, and independent assortment. Genetic variation occurs due to crossing over, recombination, and independent assortment. Right. For 2026 students, if you all didn't understand any part, you all have 3 months of access to all the recordings. So basically, after this, that means from today onwards until 3 months, usually I don't delete any recordings. So it's simply like unlimited access, but I don't want anyone to depend on the recordings, right? So you all can watch the recordings if you all didn't understand any part. Plus, you all can always ask me your doubts. I mean, not if you understand these parts, but if you have any free time, meiosis recording is a good part to be listened again, right? It's a good part to be listened again if you have time, right? If you have a free time, right? Because then it gets established in your head, right? It gets established in your head. 2025 students, study this week. Next week also study in your 35 hours of biology, right? You all can study it. Afterwards, after another 2-3 weeks also, you all can study this part, right? Now let's move on to the questions. If you have any doubts now, you can ask me, right? If you have any doubts at the moment, you all can ask me now. I will answer any doubts, right? Okay. Since you can ask me at any time, it's okay even if I'm speaking, you can ask me, right? Now, let's move on to the questions, right? 2019, there are no questions actually. There was a question on mitosis. We have done that. 2020, there is a question. I will give you no, 2020 also we have done this question actually. We did this question last week because we did this question last week in mitosis. If you were there, you might know this, right? 2021, there is a question. Do this question and send me the answer, right? I'll give you 2 minutes to attend this, to try out this question till 10:07. You all can try out this question. I'll just read it, right? 4 events of meiosis are given below. A. Centrosome move towards opposite poles, forming spindle. B. Formation of synaptonymal complex. C. Pairs of homologous chromosomes arranged on metaphase plate. D. Crossing over of chromatids. Which one of the following is the correct sequence? See, they are asking the order of occurrence of the above events. Think carefully. It's okay. Take your time. It might be your first time. So take your time, do this question and send me the answer, right? You can send me through Zoom or WhatsApp, anything you like. You can send me the answer. For events which occur in meiosis. That was the only question from meiosis and the 2022 structured essay. All the other questions were from mitosis. 2019 fifth one, 2020 fifth one, 2023 fourth question, 2019 structured essay question. All these were from mitosis. This is the only question that they have asked from meiosis. And because they have asked a major question in 2022, they didn't ask any MCQs or SCQs in 2022. So that remains as a homework. I will check the answer after I get all the three answers. So send me the answer. All the other students also send me the answer. 2 students need to send me the answer. Send me that answer. Still only one answer. Okay. It's okay. Take your time. Sometimes it might be a difficult question. Shall I discuss the question? Do you want me to discuss the question? Because still I didn't get 2 answers. I'm giving you time because it might be your first time in attempting. That's why I'm giving you time. I'll give 2 more minutes after that because this is the only question we have. Think carefully and answer this question. Done? Is the question hard or are there any difficulties? You can tell me, you can talk with me through WhatsApp. Is there any problem with the question? Okay, I'll just check the, okay, 2 answers are given. I'll stop sharing the screen to check who are the 2 students. Okay, 1 student has told 4, 1 student has told 5. Okay. 4 and 5, right? Okay. Right. Okay. Then, Sarah, do you need any time to send me the answer? It's okay. I'll give you 1 more minute just for you to attend the question so that you can attend the question. Otherwise, it will not be fair, right? Just to attempt the question. Sometimes it can be your first time in doing the question. Okay, shall I start discussing? I will start discussing, right? Okay. 4 events of meiosis are given below. Okay, so they are asking us the correct sequence. The correct sequence. So one student says 4, one student says 5. Okay, so actually these are the confusing answers because they appear to be correct, and I think according to the analysis, many people have chosen 5, and 5 is the wrong answer, 4th is the correct answer, right? Many people in 2020 A-level exam, according to the analysis, they have chosen 5, but 4th is the correct answer, right? Let's see why 4 is correct. First one, in both 4th and 5th, they are saying B, formation of the synaptonymal complex. Yes, first of all, the synaptonymal complex is the one which gets formed between the 2 homologous chromosomes, right? Then they are saying D in both the cases, crossing over of chromatids. After the synaptonymal complex forms only, the crossing over takes place between the 2 homologous chromosomes. Therefore, D is also correct. Now, the issue is between A and C. They are saying centrosome move towards opposite poles form in the spindle. Pairs of homologous chromosomes arranged on the metaphase plate. For that, you need to go to Unit 2, the book, and see, right? Unit 2, the book, and see. You can see the homologous pair move towards the metaphase plate. That is the last one, right? That is the last one which happens. Centrosomes move towards opposite poles happens before that, right? Centrosome moving towards opposite poles happens before that. I think, which question? 2021, right? Yeah, 2021. So basically what happens first? A should happen first, right? Centrosome move towards the opposite poles. Then only the pair of homologous chromosome arrange on the metaphase plate. So B, D, A, C answer is answer number 4, right? Okay, my screen is not been shared. I'm sorry for that. Answer is B, D, A, C. Answer is B. I'll explain again quickly. So you can see first the synaptonymal complex takes place. First the synaptonymal complex forms. Then the crossing over takes place. Then in Unit 2, you can see here they have said at last, the 2 homologous pairs then move towards the metaphase plate. This happens at the end. You can see before that only the centrosome move towards the opposite poles. So this is the 3rd one. This is the 4th one, right? So basically pairs of C will be the 4th one and A will be the 3rd one, right? That is the reason I told you. These usually the biology is not a hard subject to be studied. Biology is very easy. They make it difficult using the way, that means they make it difficult by asking us to write exactly what is in DRP, and by making it difficult by asking us to study the order. Who would have studied this order and go for the 2020 exam? Who would have done that? Only like 1%, right? From everyone, because most of the students have got the 2024 question wrong. But that doesn't mean you will lose an A because of that, right? You still can go for, you still can get 49 out of 50, right? Even if you miss this part, right? Because of the, because you haven't studied the particular order, right? So basically, after 2021, after looking at 2021 paper only, I understood, okay, then I need to study the order in both mitosis and meiosis, and I should apply it for all the other cases where there are processes. In the future, if I find any process, I usually study the specific order as well, right? Then I won't ever face this sort of situation like the 2020 students faced. Because usually we are asked to study what happens in prophase, what happens in metaphase. If we were asked to study like that, it's very easy, right? But the hard part is to study it in a specific order, right? That's why I told you, you can't keep the order in meiosis 2 part. That means you sometimes can confuse, but you have to keep it in your mind. So to prevent that, to prevent from getting confused on it before all the model papers, you should just read that part and come, right? You should just read that part and come, like understand the orders and come before all the model papers. Sometimes in the model paper you won't be asked, but you have 100% confidence level to do the paper when you have studied everything, right? Okay, so this marks the end of the free webinar, and I will send the recording. I will upload the recording to YouTube so that everyone can watch it, right? And thank you for joining, and thank you for waiting until the end of the class. Do the homework that I'm sending you. Everyone, you all will receive this weekly bundle this week. The Meiosis weekly bundle this week itself. No, I think for 2026 students, Meiosis weekly bundle, it depends. If I can't remember whether I gave you the Mitosis weekly bundle. I think I didn't. So first I'll give you the Mitosis weekly bundle. Then I'll go for the Meiosis one, right? Okay, so 2025 official students will also receive that. You're welcome. Now, and I will end the class. Thank you for joining. Okay, right.